The adaptor hypothesis was first proposed by Francis Crick, originally in a privately circulated note in 1955 and published later in 1958. He suggested that nucleic acids which interact by base pairing through hydrogen bonds were unlikely to be able to distinguish between the different amino acids, especially those that differed by only one methyl group. He therefore proposed that genetic messages would not read amino acids directly, but that each amino acid would be linked to an adapter molecule, probably a small nucleic acid with 20 enzymes to perform the specific linkages. Thus, whereas nucleic acids could not easily differentiate between the 20 amino acids, a protein could, by recognizing both, specifically join the amino acid to its adapter, and the adapter could then be recognized by the message by standard base pairing rules. Although very much later it was shown that nucleic acids could be themselves recognize a wide range of molecular configurations by adopting three-dimensional structures, the hypothesis was enormously prescient, predicting as it did the existence of transfer RNAs or tRNAs and the tRNA amino acylases. At the time, however, its main impact was the realization that the degeneracy of the code need not follow logical rules, but could simply be due to historical accidents which assigned the triplets to the different amino acids. Adenine. Adenine is one of the purine bases found in nucleic acids. When attached to ribose, it is the nucleoside adenosine. When attached to deoxyribose, it is the nucleoside deoxyadenosine. The phosphate esters of those nucleosides are the nucleotides adenylic acid, adenosine plus phosphate, AMP, and deoxyadenylic acid, deoxyadenosine plus phosphate, DAMP. The triphosphate forms of adenosine or ATP and deoxyadenosine DATP are substrates for the synthesis of RNA and DNA respectively. When present in RNA and DNA, adenine functions as one of the letters of the genetic code. ATP is a ubiquitous high-energy substrate and, along with GTP, a cofactor in many cellular reactions. Adenoma. An adenoma is a benign neoplasm arising from glandular or secretory epithelium. It is classified by reference to the cell lineage of origin. Thus, a benign neoplasm arising from the glandular cells of colonic mucosa is a colonic adenoma. An adenoma arising from secretory epithelium of thyroid follicles is a thyroid follicular adenoma. Occasionally, the term may be further qualified to reflect macroscopic features such as cyst formation, for example, cystadenoma of ovary. In some circumstances, adenoma may progress to a malignant neoplasm termed adenocarcinoma. Before this happens, the adenoma cells may show evidence of disordered maturation and growth known as dysplasia. Thus, the complete designation of an adenoma contains reference to site, cell type of origin, and degree of dysplasia.